Okay, so what I'm showing you here is the actual slide from your PowerPoint. The next experiment that we did in lab was using the enzyme Catalase. The source of that enzyme was potato. So there's catalase within the potato cell. So again, how we recognize that term catalase as being an enzyme is that it ends in ase, A-S-E. Catalase's substrate is hydrogen peroxide. So here's our reaction. Hydrogen peroxide serves as our substrate, catalase is our enzyme, and when the reaction takes place, the products will be water and oxygen. So what we did was we asked you to put hydrogen peroxide into your tube or your cup, and then you added potato, which was your source of catalase. And then if the reaction took place, what you visually saw was the liberation of that oxygen. So it was the oxygen that you saw forming the bubbles in the solution. And so it kind of looked like something like that. That's a picture of a test tube. But the catalase reaction, hydrogen peroxide is liberating oxygen, and you get all of this bubbling going on up here. So what we did, again, was you looked at a couple different things. You looked at heating the potato, and then you looked at adding more concentration. So the first thing we did was we used an unheated potato versus a heated potato. And we saw what kind of observations we got. With the unheated potato, you should have gotten nice effervescence or bubbling, meaning that the reaction did take place. With the heated potato, you should not have gotten much at all, if any. And that's because when you heat an enzyme, you are denaturing that enzyme. What happens when you heat the enzyme is that you increase the molecular movement because you're adding heat, and that's enough to break bonds between the amino acids of that protein. And so it unfolds, its shape goes away. And remember how we talked about, I'm going to swoop you over here real quick, the enzyme actually having a particular shape, right? So if we break the bonds in here and now it doesn't have its shape anymore, it comes loose, it won't have those active sites for that substrate to bind to. So a denatured enzyme might go from something like that to something like, well, there's our big enzyme now, and it's obviously not going to work because it doesn't have its original shape for those active sites. So when you heat an enzyme, you denature it by breaking the chemical bonds. Denaturing it means that you change the shape and you destroy the enzyme. In other words, you render it inactive. It will no longer work. Drastically changing the pH of the solution will also do that, but we didn't look at that in lab. We just looked at adding heat. So those are the results you should have had with that one. Now, with the next one, flip over here, we asked you to add more potato to different tubes. So we only had 3 tubes. This picture I found has 4. But you had a tube, you had tube 1 with 1 piece of potato, tube 2 with 2 pieces, and tube 3 with 3 pieces. And you should have seen an increase in the rate of the reaction, just like what you're seeing here in these 3 tubes, where we have a small rate of reaction, a greater rate of reaction, greater rate of reaction, as measured by an increase in the little column of bubbles that you see there. The reason that you get an increase in that rate of reaction by increasing the cubes of potato is because when you increase the potato, you're increasing the concentration of the catalase. So the higher the concentration of the catalase, the better reaction you should have gotten in that tube.